So let's get into innate immunity, and what we will go through in sort of in this order is the physical barriers we have, some chemical barriers, and then we will look at the behaviors of the cells of the innate immune system. So we'll look at some major things that those cells do. So let's look at that. So the best part of our innate immune system, the thing that keeps us alive, that saves our life every single day, is our skin. And so if you can picture your epidermis, the most superficial part up here will be colonized by bacteria. And so our special microbiota, like our normal resident microbiota, will be there, but also potentially anything else, including deadly pathogens. And so when the superficial layer sloughs off, that protects us from them. They fall away with that skin cell, and they're no longer on us. We also have mucous membranes that can protect inner surfaces. And by pushing the mucus along by a cilia, a similar process happens where we move invaders from sensitive areas such as our lungs to safer areas such as our stomach. So let's see. There are a lot of different chemicals involved in innate immunity. A lot of them are just antibacterial, so there are a lot of different enzymes, but the ones that are most famous would be like lysozyme, and that destroys peptidoglycan. And in the next slide, we'll look at sweat. Sorry. What I would say about things like this, whenever I show you a list, if I'm only showing you on one slide, it's probably not the most important thing to study. I will show you some tables later, and I never want you to memorize a table unless I tell you to do it specifically. So a lot of times I'll be showing you these lists to jog your memory, but I won't likely test you on things I only show you once. So that was neither here nor there, I guess, but we have to have both antibacterial and antiviral chemicals, and we have to be protected against these things all the time. So as I mentioned, sweat, and when sweat evaporates, it leaves salt behind on the surface of skin, and that drives down the water activity of skin. And so this table you've seen several times now, where we have a water activity of pure water, and so that readily moves into cells. But if we go down to like seawater, that would draw water out of typical cells. So these cells have to be adapted differently for those water activities. And bacteria up here mostly would not be able to grow in lower water activities like seawater or bread. Definitely around 0.9, most bacteria, most fungi can't grow on the surface of skin just because it's salty and too dry. And so some bacteria that can, and some fungi that can, well, we see them there because they have an advantage. So Staphylococcus is an example of something that can grow in that salty, drier, more dry environment. And whenever people live in a humid place, they're more susceptible to skin infections from fungi. So we saw cutaneous mycosis or dermatophytes. Those infect people in parts of their body that have more moisture, and those are more common in parts of the world that are more humid. Okay, so I really didn't want to spend that much time on the chemical defenses but the cellular defenses definitely. So phagocytosis is a very central part of the whole immune system. It's an innate function done by many different cells, and it's how we can initiate immune responses. It's also how we can kill invaders. So in phagocytosis, what a cell does is it puts its cytoplasm, it kind of deforms itself so that it projects around the invader, and that allows it to pull in the invader through a process kind of like endocytosis. I mean, it is a form of endocytosis, but it's endocytosis of a large object. And so the cell has to kind of deform itself around the object. And then, so if we're looking at a bacterial cell, the bacterial will end up inside a vacuole, and that's called the phagosome. The cell also has vacuoles called lysosomes that are full of digestive enzymes, and those will fuse with a phagosome to make the phagolysosome. That is a very dangerous place for bacteria. The whole purpose of that is to digest this cell and turn it into its parts. And then those can be released or processed and displayed, depending on what kind of cell this is. So, why do cells do phagocytosis? Well, it cleans our tissues. It can physically remove cells from our tissues, and it can kill invaders. And so, for example, macrophages are constantly able to do this. They roam through our tissues, cleaning up debris through phagocytosis and recycling it into molecules that the cells can start to reuse. So that's part of the purpose. It's taking like a dead part of a cell and breaking it down into molecules that can be reused. In other cases, phagocytosis is kind of the eyes and ears of our immune system. So dendritic cells, for example, will do phagocytosis and other similar things like pinocytosis in order to find pathogens and present them to the adaptive immune system to sort of ask, hey, is this thing a problem? So dendritic cells are going to do phagocytosis in order to sample the environment. Macrophages can do that too, but they more are going to ask the adaptive immune system, I found this thing. Are we currently mounting an immune response against this thing? And if so, the macrophage will activate and it will change its behavior. But both of those processes require phagocytosis. And so phagocytosis is a step that can lead to antigen presentation or just killing of the organism. This really is a fantastic thing that our cells are able to do to protect us against bacteria. If you then go and look at real pathogens, famous pathogens that can make a healthy adult sick, what we see is ways they avoid death through phagocytosis. Lots of different pathogens avoid phagocytosis in different ways. So some of them just live in parts of the body, or they infect parts of the body that don't have phagocytes. So Trichomonas, for example, will not penetrate a tissue. It lives in the mucosa. Others will disguise themselves as self so that phagocytes can't recognize them and don't know to phagocytose them. Still others make it physically challenging for phagocytes to phagocytose them. Others will interfere with the process of phagocytosis down later. Like these, like chlamydia, legionella, mycobacterium, will allow themselves to be phagocytosed, but they won't allow the macrophage or whatever phagocytic cell it is to fuse lysozymes to the phagosome. And that means the cell that sort of ate these bacteria, so to speak, can't kill them. Another way bacteria get around lysozyme or lysosomal killing them is just escaping from the phagosome. And then the craziest thing is, like, Staph aureus will just survive the onslaught of enzymatic and oxidative attacks from the lysosome. So these are all pathogens that can kill a healthy adult under some circumstances, and they all have to have ways of dealing with phagocytosis. I'm not going to ask you to, I'm not going to test you on this slide, but I will absolutely expect you to know that pathogens have to be able to do this. Oh, and I forgot to tell you, some of them hunt phagocytes and kill them. Some of them live in phagocytes. I couldn't find the examples of these, but some of them will actively find phagocytes and live in their cytoplasm. Others will actively kill them. So, macrophages are our primary phagocytes because they're in the tissues all the time, cleaning up the tissues and removing debris when no infection is happening. But during an infection, they will be very effective at phagocytosing pathogens. They also have the ability to present antigens. As I mentioned before, they can do that for two different reasons. Neutrophils are other cells of the innate immune system, and they are specialized to doing phagocytosis during infections. That's really all that they would do. So they're not going to be cleaning up tissues the way macrophages would. They are going to specialize in fighting off. Good enough. Dendritic cells, I mentioned, are kind of the eyes and ears of the adaptive immune system, and they are going to do phagocytosis in order to present antigens to T cells, to sort of ask the T cell if it's time for it to start a major immune response. So dendritic cells will live near epithelia so that they can sample the contents on the other side of the epithelial layer. Yeah. And yes, so once they have collected enough antigens, so to speak, or once they've collected enough cells or debris or whatever, they will migrate to lymph nodes, because that's where the T cells are waiting to be activated. So if you look at a T cell under enough magnification, you'll see dendritic cells wandering around, presenting antigens to all the different T cells as fast as possible. There are a few more phagocytic cells that are specific to organs, and less is known about these. But you probably learned about microglia when you were learning the nervous system. Microglia are the phagocytes of the central nervous system. They have a strange role in the body because the blood-brain barrier, or the blood-CSF barrier, really prevents things like antibodies from getting to them. So they don't get to, looking ahead a little bit, they don't get to look for opsonized pathogens. They have to have like more direct ways of interacting with T cells. I was looking at sort of what their functions are because I can't find any details about them in my immunology books. And so the papers I was looking up that were showing me kind of what they do are all from the last decade. So this is an active area of research. I don't think anyone is very confident about how microglia do their jobs. I don't know how to say these cells, but these are the macrophages of the liver, and they are very numerous. They live in the sinusoids, And so they are patrolling the blood that comes from the gut and is going to be purified by the liver. So they do a big part of purification of blood in the liver. As phagocytes go, these are the most abundant tissue phagocytes we have. Technically, they're macrophages, so they're the most abundant tissue macrophages we have. That is, if you took all the tissues in your body outside the liver and all the tissues in your liver and compared them, you'd find more macrophages in the liver than in all the rest of the body put together. So excluding monocytes in the blood. And dysfunction in microglia or Kupffer cells, I assume is how you say it, they're both dangerous, but in both cases, immunology books don't even mention them. And they get brief mentions in my physiology books, but I think they're both a little bit mysterious and still slowly being understood. So here's a list of phagocytic cells. You should know their major functions. You should definitely know their major functions because they are different from each other. These are very specialized cells. And I'm going to stop this video here and come back and look at how phagocytes know when they should do phagocytosis. So I'll see you there.